Hi everyone. I'm really glad to be here today, and I would like to sharing with you a part of my PhD work. So, under the title of SARS-CoV-2 MERS envelope protein interactions. At the beginning, as a brief introduction, the MERS coronavirus and the SARS-CoV-2 coronavirus cause the respiratory illnesses with potentially fatal outcomes, as we all know. Both MERS and SARS-CoV-2 are beta coronaviruses, and they are single-strand enveloped RNA and encode 4 structural protein, envelope, nucleocapsid, spike, and membrane proteins. To date, most antiviral act by directly targeting viral proteins, which actually can be mutated and resulting in drug resistance. Viruses have significantly less coding space in their genomes, and because of this, they rely on a host protein to supplement the activities of their proteins. Targeting host proteins required for viral replication can avoid resistance and develop broad spectrum of therapeutics. Indeed, families of viruses frequently share cellular pathways, processes, and coding component capable of modulating cellular signaling pathway and shaping their distant immune signature. Most coronavirus, SARS-CoV-2, SARS, and MERS coronavirus proteins shown the same localization pattern supporting the previous hypothesis. It is necessary to characterize the host targets of coronaviruses to better understanding their roles during the virus life cycle. I was talking about a little bit of systematic because it's an introduction. So now my project's goal, or the main project's goal for this presentation or this part of my work, is using a high throughput quantitative proteomics to study the MERS and SARS-CoV-2 E proteins interactions in 3 different types of cells. So we used human, bat, and camel cell to identify the host protein that may modulate the coronavirus replication and could provide a new antiviral targets. Of course, we use a bat because it's known as an animal reservoir, as we all know, for coronaviruses, and the camel because it's known an intermediate host for the MERS coronavirus. I will go through now a little bit specific. So I have 3 specific projects goals, which is transiently express the MERS coronavirus and SARS-CoV-2 genes in selected human, bat, and camel cells, and perform a high throughput interactomic analysis using a mass spectrometry. This was the first one. The second one is a bioinformatically compare the human, bat, and camel cell dataset to identify a highly conserved interaction for these 2 coronaviral protein. Oops. The third one, confirm the relevance of selected interaction by depleting the host proteins of interest by using the interfering RNA, which is a small interfering RNA, which they call it siRNA knockdown. So I use 3 different types of cells, which is human, HEC293 cell, and bat, or they call it bucket, and Dubka cell from a camel. Cells were used for transient expression to produce both MERS and SARS-CoV-2 E proteins, which is a flagged epitope tagged. Cell transfection was done for 24 hours. This is a diagram that can represent and shows how the co-immune precipitation was done briefly and quickly. So the first step, the cells were grown, transfected, harvested, and lysed. After that, the cell lysate was added in a new fresh tube. And this actually sample should include the protein of interest, which is a viral flag tagged protein, and another cellular protein interactor. We take a little bit of these samples and we call it input sample. After that, the cell lysate was incubated with the anti-flag magnetic beads for 24 hours. So these magnetic beads, the anti-flag should interact with the protein of interest, which is a viral protein in our case, that should interact with other cellular interactor. After the incubation, we use a magnetic rack to collect the beads, and we take a little bit of supernated as a non-bound sample. After that, the beads was washed and the pull-down sample was ready. After we prepare the pull-down sample, we take this pull-down sample and send it to proteomic analysis. And yes, this is the first part. The second part, we use input, non-bound, and pull-down sample to do a Western blot, just to make sure that the viral protein have been produced in the cell, in the transfected cell. So as it shows here, so this is the cell lysates from the cells were transfected with the MERS coronavirus. So this is the input sample, non-bound, and pull-down sample. As we saw here, the envelope protein was shown in an input and pull-down, which is expected, and not in a non-bound. And the same in the SARS-CoV-2. This was shown, let's say, this is cell lysate from one of the cells that we used. So for example, this shown a HIC Western blot, and we have the same for packet and for Dupka cell. The pull-down sample, when it was sent to the proteomic analysis, we end up by having 6 data sets. We have, as I said from the beginning, we have 3 different types of cell. When we multiply it by 2 viral protein, we have 6 data sets. So here, HIC293 cells, when it was transfected with SARS-CoV-2 envelope protein, we end up by having a list that should introduce the cellular interactor, which is, we hypothesize that this cellular interactor is really important for the SARS-CoV-2 envelope protein. And it has the same story for the other data set. So we achieved the first goal. The second one was bioinformatically compare the human, bat, and camel cell data set to identify a high conserved interaction for MERS and SARS-CoV-2 protein. So as we mentioned, we have 6 data sets. These data sets need to be analyzed and to understand and select most, if I can say, interesting cellular interactor. So we don't have a problem with the HIC293 cells because the protein's name is in a human unknown, but we need to do an annotation for the packets and Dupka data sets. So we did this by using String, Uniprot, and Galaxy website. So after that, we use statistical tools. So we select the protein that has a log-fold change more than 0, and the t-test should be less than 0.05. So we identify now these the number of proteins on each data set that follow the criteria. After that, I use a filter. I call it both viruses. So both E protein, the MERS E protein and SARS-CoV-2 E protein on each cell. So we have, let's say, HIC293 E dataset that represent the shared cellular interactor for the MERS E and SARS-CoV-2 E in a HIC293 cell. So out of 6 datasets, we end up by having 3 datasets that represent each different type of cell. These 3 datasets, I use it to do a compare by using a FunRich website. So this is the diagrams. This will show on the protein, which is a packet E dataset, and here the HIC, which is a human E dataset, and the Dupka, which is a camel dataset. Out of these 3 datasets, I found that there is 21 cellular interactor, which were shared between these 3 different datasets, which is, let's say, we can call it a promising cellular interactor that we can use it in the next step. So we achieved the second goal. The third one, which is the last one, confirm the relevance of the selected interactions, this is the 21 protein, by using the siRNA knockdown technique. To do the siRNA knockdown, these 21 proteins were depleted by 4 pooled siRNAs knockdown in an A549A2T2 cell in a 96-well format. So I use this diagram as an example. So we have UBA52 protein. The siRNA's transfection was done for 48 hours. 2 levels of multiplicity of infection were used, low MOI and high MOI. 3 independent experiments and 3 technical repeats were done. So let's say, for example, we have this UBA52 protein. Each plate introduced one experiment. So in a low MOI level, we have 3 different experiments, and each experiment has 3 technical repeats. So in the total, let's say for the UBA52 protein, we have 6 different experiments, 3 low and 3 high MOI. To introduce what we did exactly in siRNA knockdown, the A549A2T2 cells transfected with the siRNA for 48 hours, as we mentioned. Then we infect these cells with the SARS-CoV-2 for almost 24 hours. After that, cells were fixed, stained, probed with an antibody against the SARS-CoV-2 N protein, and analyzed with an image express pico machine. Out of these 21 cellular interactors, we found one which is shown an interesting result, and I just mentioned it in a few slides ago, which is the UBA52 protein. So when this protein have been knocked down in the experiment of this protein, we can see the percent of SARS-CoV-2 infected cells were almost reduced about 60% in a low MOI experiment, while in a high MOI experiment, the percent of infected cells were almost reduced by 20%. This picture was taken from an express pico. This can show the control cells, and actually the orange color can represent the infected cell, while when UBA52 was knocked down, we can see the cells here. Most of it hasn't been infected. So it seems like the UBA52 give us an interesting result. So I just go back. Woo! Give me a minute. I'm almost finished. So it sounds like the UBA52 shows a promising result. So I just figure out or try to find other articles. I found this one and give us a quick information about UBA52. It's a fusion protein consisting of ubiquitin at the N-terminus and the ribosomal protein L40 at the C-terminus. UBA52 was identified in these articles as an influenza A virus host protein interactor using chicken embryonic fibroblast cells. The H5N1 titer significantly decreased after the UBA52 knockdown. In other articles, quickly, it talks about the ubiquitin proteasome system plays an important role during various stages of coronavirus infection cells. And they mentioned that the MHV replication was reduced when the UBA52 was knocked down. As a conclusion, and for future work, this study has investigated the interaction cellular protein that appear important for SARS-CoV-2 replication, including the UBA52. These findings can follow up by investigating whether drugs can safely target this protein to reduce the viral replication in the cells. And at the end, because of the time, I just highlight the project's guidelines. If you are interested for more information, I'm happy to help. At the end, I would like, by the way, it's a quick slides. I just made it quickly. So I would like to thank the Ministry of Health in Saudi Arabia who gave me a chance and the grant to do my PhD. And a big thanks for the University of Bristol and to my supervisor, Andrew Davidson, and for all his group and my colleagues there. And the big thanks for the UK-ICN to give me the opportunity to introduce my work. And thank you for listening.
Any questions? David? I saw David's hand go up.
Thanks. I was wondering about your high versus low MOI, because I noticed it was only a 5-fold difference between them, so less than 1 log difference. So when I think high and low MOI, I think MOI of 1 and MOI of 0.01 at least. But you see quite a difference in your phenotype. Do you think if you moved higher or lower, that dynamic range would disappear? Would you still see an effect?
It's an interesting question, I think, but I don't know exactly, to be honest. Yeah, it's a tricky point. Yep.
One more quick question from the back.
I noticed that your interactions with the camel cell line,
Yeah.
you didn't get very many. Is there a reason why you didn't get so many of those?
Maybe there's more than one reason. One of them is, let's say, when we annotate the data set, maybe there is something missing there, because at the end we try to annotate it and give it, if you can say that name, more or less like human. So maybe some of them was missing. And also, I think maybe there is no a real interactor in a camel cell in comparing with a human. This is the points that maybe I can think about it now. I hope this could help.
Okay, thank you very much. We should move on.